Yo, what's up? Yeah, it's me. Okay, I'm back. Yeah, I know I skipped a day. I thought I was gonna do this, on a daily basis. I'm in a race here, man. I'm telling you, in a race. Okay, so I've got a lot to do before I can really figure this out. This secret of eternal life. There's just a lot of and the people who study biology and chemistry, if there's any PhDs who are watching at this and maybe laughing a little, you probably already are like, you're asking me, like, why didn't I get a biology degree? Why didn't I start grokking this years ago? If I was really interested. Well, I didn't really know how interested I was in this until I realized that, the universe is so crazy huge. Okay, I'm gonna go on a slight tangent, but it's not really. The universe is so crazy huge. Okay, like, I don't know what the observable size of the universe is, 93 billion light years or something just incredibly unfathomable to the imagination. Okay, and there are so many secrets to the universe. And the sad thing is, I thought about this actually 2 years ago. I was watching astronomy and videos, and I wanted to really get into physics. And I saw a series of videos about the superscalar structure of the universe, right? So what is the superscalar structure of the universe? That's just how the universe is mostly comprised of dark matter, but there's a skeleton of galaxies and super galaxies that comprise the, I guess, backbone of the universe. And this multi-billion light year long, quintillion mile, hexillion mile long universal landscape is filled with secrets that I think that in a lifetime man will never know. Okay, the only way to know these secrets is to be the Ponce de Leon. We gotta get to him. We gotta get to that point. We can't, I can't. I just can't go through life. I have many reasons to be on this journey, but I just can't go through life knowing that there's all this I'll never know about. I've got to know about it. Okay, I feel that in my several years, my multiple decades of being on this planet, this was something one of my purposes. So it's really like this is a fundamental thing, and there's not a lot of time left, frankly. I've got to really start grokking this, and these videos I think they're going to help me, but it's kind of sad because it's like, I devoted a little bit of time each day to understanding the science, but I've been realizing that, I need more and more time. I just need more and more time every day to really grind on it. And it's really tough because I have so many other responsibilities, but to master biology to the point, and biochemistry and genetics to the point where you can really start to, for yourself, because I've always believed in doing many things for myself, and I truly believe in my heart of hearts the only way that I'm going to get to this point in time, it's to figure this out, is in a large part by myself. Now, there are universities, people spending billions of dollars to research this every day, people making miraculous breakthroughs in all realms of biology constantly. It's just unfathomable to how much is being discovered. But this fundamental goal, right? This fundamental goal, which I should have realized a long time ago was a fundamental goal of mine, really requires study to an extent that I need to commit myself more to, like, with the religious zeal. So I've given myself like about a year of time to understand to really concretize in my mind what would be the fundamental principles of biology up to what I would say be a PhD level. Now, I intend to spend 3 hours a day doing this for a year, okay, at least. And it's tough because compounding the difficulties of biology, organic chemistry come along, and they both create this really huge just huge, very, very difficult science to surmount, because it's actually real life. Reality is very difficult to surmount. Okay, I mean, reality is science. The science of reality, just living every day, day to day. These are very difficult things, and it requires sometimes a sacrifice of proportions that it takes you to get to a certain point to realize that you must learn this. I don't think when I was younger, I really had it in my mindset that I must. I did not have the fire under me to feel that I must learn this. But now I feel in order to get to the eventual ultimate goal, and then several other intermediate goals, I have to learn this stuff. You know, I really have to hammer this down. But it's fine. I have my own ways in my mind of creating shortcuts for myself. I have a very, I think, a daring plan to get to where I need to be, but I know I have to get through this first. So anyway, I've been a little abstract with what I've been talking to you about, because it's really important, right? Like I've got to really go through the rigors of what a biology student goes through, like a real biology student. And the thing is, the kind of sad thing is that I've taken biology in school before. You know, I remember at some point learning a little bit about all of these topics, but never to the extent that I cared so much about it until now. And you know what? I don't think I could have actually gotten to the point where I did when I was younger, care about it that so much as I do until now. I tried. It's just that I didn't have the fundamental impetus of a limited lifespan that I have in front of me as I speak when I was younger to really appreciate the urgency of this endeavor. So let's start with that. That's my long introduction. So let's start. Okay, so glycolysis. You all know what glycolysis is, right? Glycolysis is really the most basic pathway of energy production in any living thing, okay? Pretty much anything that's alive goes through glycolysis. And I'm going to be jumping around all of this because I'm really also learning the deep details of glycolysis myself. But anything alive, bacteria, pretty much, and someone can correct me if they want, but anything alive goes through taking, at the very start of their respiratory life cycle maintenance process, where they're keeping themselves alive, they start by taking in a glucose molecule and converting it to pyruvate. And that pyruvate either goes through a more complex process called cellular respiration through the Krebs cycle and pyruvate oxidation and electron transport chain, or it goes through lactic acid fermentation or alcohol fermentation, depending on what it is that's happening, what life form it is that is trying to create that energy. So fermentation is basically, and again, this is a recap for many people. So many people probably already have, know what this is, and you learned this a little bit in high school, though I doubt most people really appreciate it in high school, except in the context of educational television programming that tries to throw in, these terms, whatever, yada, yada, very big deal in the 90s. But glycolysis, fermentation is, basically it's the waste process of glycolysis, right? It's like, well, if the organism can't actually go through with a more complex, more efficient process of energy production in the form of ATP generation that the Krebs cycle allows you to go through, then it will go through fermentation. Okay, and people go through fermentation too. People need a lot of energy normally, right? And so here's the deal. Like glycolysis is a simple process. By itself, it doesn't require any input oxygen to operate, right? That's one of the nice things, because way back when the Earth was first, filled with life that wasn't oxygen consuming, I think we might have gone through that at some point in time. When most of the atmosphere wasn't oxygen, there were things that took in reagent molecules and produced oxygen. Eventually, the atmosphere got filled with oxygen, and then living things started using oxygen to produce energy because it became way more efficient to do so. But before all that, and when I mean oxygen, I mean molecular oxygen, okay? But before that, there were molecules, glucose back in the day when it was around, was taken in, and taken in by things that were pretty much like mitochondria, the first primitive bacteria which endosymbiotically bounded to our cells through phagocytosis at some point, and then instead of dying, combined with our cells and became the powerhouse of our cells, right? So that they are dependent on our cells and our cells are dependent on them, okay? So anyway, glycolysis, the mitochondria do a lot of things. They are responsible for the bulk of energy generation in the cells, but pretty much all cells still retain the ability to do glycolysis without mitochondria. Glycolysis occurs in the cytosol of a cell. So somewhere within the cytosol, probably several places within the cytosol, and the cytosol, remember, is the liquid portion of the cell, you have glycolysis happening, okay? Now, as glycolysis happens there, it goes through, there's several steps. Glycolysis, it produces only 2 ATP. It also produces 2 NADHs, right? And it also produces 2 ATP, and it produces 2 pyruvates, which go to fermentation, right? Now, the 2 ATPs, a very, very tiny bit of energy. Remember, ATP is, and it's popularly mentioned in textbooks. I've talked about it before here. ATP is the energy currency of cells, okay? So what does that mean, that ATP is the energy currency? Well, why not energy? Why isn't energy the energy currency of cells, right? Like, what is energy? That's one of the fundamental questions that you will need to live long enough, which means probably infinitely long, or not infinitely long, but maybe several hundred thousands of years to really, truly understand from this point on what energy is. But at this point, we know that energy, is the motive substance of everything, right? Things need energy. And energy can be transferred. It can't be created or destroyed, but it can be transferred. And matter and energy are equivalent. Einstein's special relativity theory, E equals mc. Let's find an image of that, because I need to start talking, showing rather than talking. E equals mc squared, right? Which is pretty much what the sun does all day, every day. Is energy, right? And we have M and we have C, but we can't C being the speed of light. We can't just do E equals MC. You can't just take M and C, multiply them and get E, because to do that requires processes beyond the capabilities of cells. There are no cells that are capable of nuclear fission, okay? Or fusion, or any of the things that directly convert E to C. So the best you can do is transfer via stable and unstable molecules some E, okay? And ATP has some E within its bonds, okay? ATP and has E stored in the unstable third phosphate. Remember, ATP is, I gotta go up JS Paint again. Let's do this. Remember, ATP, I've drawn ATP before. ATP, don't ever allow data access. ATP consists of phosphates attached to each other, right? So phosphates look like this, right, etc., 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 etc. And then you have one more phosphate, and then you have the rest of the ATP molecule, which is actually not that complicated, but let's just look at these. So ATP contains these phosphates. ATP, by the way, is a primordial molecule. You know that because of the A, okay, adenosine, adenine, right? And the A stands for adenosine because it's a adenine-based ribose sugar, but the adenine is a primordial amino acid that exists in everything, and it's pretty much peppered throughout all of biological molecules in different motifs in different places. But anyway, even NAD, right, has adenine in it. But anyway, so P is stripped off, right? This is a high energy. ATP is a high energy. This is a high energy bond, and it wants to release its energy. It wants to go to a lower version called ADP, right? ADP. And what happens through many reactions, called specifically phosphorylation reactions. That's how phosphorylation, you can think of ATP as the currency, you can think of phosphorylation as the transfer mechanism. So when I give you money, I'm giving you a phosphate, right? And to do stuff with, and you know, the ATP is the result. It's not exactly that an analogy of change to money, but you might be thinking change, but any ADP is the result of, is the successful transaction of a phosphate is the result of a successful transaction of a phosphate from an ATP over to whatever mechanism that needs to run. For example, I'm going to get into this, a lot of the molecular motors, all of the molecular motors in your body pretty much, for the most part, use ATP or GTP or some kind of, phosphate loaning mechanism to do their activity. Once phosphate is attached, the phosphate motivates an action. So, what implication does that have for glycolysis? Okay, so ATP is produced in glycolysis. ATP, once it's produced, it can be used pretty much anywhere in the cell, right? Your cell uses ATP everywhere to run its sodium pumps, right? So it can pump sodium out of a molecule and take in potassium. It uses ATP even to produce its own self. It uses ATP, right? So when ATP synthase works, it works through a proton gradient that is, essentially driven through ATP. I'm not going to go into that right now because that's something I really got to dig into myself. That's part of the electron transport chain. That's what really, quote, advanced organisms do to generate energy after the Krebs cycle. But let's say for now we're still on the very basic organisms that use glycolysis. That ATP is used again in other glycolysis reactions, and so on and so forth. Glucose taken in, ATP produced, ATP used, and so on and so forth. The ADP recycled, and ATP is rephosphorylated back onto ADP to create money again, and so on and so forth. So basically, this is how glycolysis behaves. Now, glycolysis produces ATP, and glycolysis produces NADH. Okay, NADH, interesting thing. A lot of people call NAD the miracle molecule. A lot of these, by the way, a lot of the molecules I'm talking about, like nicotine, adenine, dinucleotide, NAD. By the way, look at NAD. Okay, let's take a look at NAD. NAD is a beautiful, in the words of Donald Trump, he said this exactly. No, I didn't. He did not. I can't misquote the President of the United States. But you can imagine him saying this: nicotine, amide, adenine, dinucleotide. Okay, nicotine, this here, right at the nicotine, okay, nicotine amide, adenine, dinucleotide. This is the nucleotide. There are two of them, so it's dinucleotide. Remember, a nucleotide consists of what? A phosphate backbone, pentose sugar, and in the form of a ribose, and amino acid base. Okay, in this case, the amino acid is nicotinic acid, and it's nicotinamide, right? In this case, your amino acid base is this guy here, which is adenine. Okay, adenine is glycosidically bonded through an N-glycosidic bond to this pentose sugar, and then this pentose sugar is bonded through its 5' carbon, right, to 5' carbon to another nucleotide. So anyway, NAD, right? Let's talk, before we even talk about NADH. NAD is one of the most important molecules in cells, as people like to call it, okay? It's used to transport electrons in cells. And the way it's used to transport electrons in cells, NAD gets oxidized. It gets reduced, meaning that an electron gets added to it. How does an electron get added to it? Well, a hydrogen gets added to the electron, gets added to the NAD, and therefore you have NADH, right? NADH means it carries an electron, and actually it carries 2 electrons. NAD plus is what you start off with, right? And then it becomes, it gets an electron, so it becomes NAD, then it gets another hydrogen attached with an electron, so it becomes NADH. So essentially, it's what people like to say, an hydride transfer to the NAD. Okay, anyway, that hydrogen, so NADH will be carried over to other places, specifically to your mitochondria, where it can go through oxidative phosphorylation, and it will send in the hydrogen through the proton gradient of the mitochondria and back into the mitochondria through the ATP synthase, creating. Anyway, that's a lot of stuff there. I know this is a lot, and this is a half-an-hour video, and I'm going through everything, okay? I'm going to break it down, and I'm going to get better at doing these videos, but you know what? Got to start somewhere. Okay, it's not half an hour, it's going to be 25 minutes. But anyway, there is a lot that goes on with NAD, and people like to market NAD. But we'll go back to it in glycolysis, okay? Glycolysis is the important reaction to talk about here, or a set of 10 reactions, rather. Glycolysis. Remember, okay, and then when we're talking about reactions, it's good to remember what's the point. You start off with something, you end up with something. The thing you start off with is your reagent. The thing you end up with is your product, and in the middle you do a bunch of other things and have a bunch of other intermediates, and you generate a bunch of other things like NAD and ATP, which are used for all those purposes. You want to generate energy, and then then because life is very efficient, strangely enough, you might think it's dumb, but it's efficient. It does other things with the downstream pyruvates that are generated. Okay, anyway, that's enough for now. I might get a little deeper into glycolysis. Next time I might talk about protein pumps. I'm going to really jump around until I can present to you my day-to-day coherent structure of life, and then be able to talk about modern research, okay? Because that's really what's important. This has all been discovered 50, 60, 70, 80 years ago, longer than that. Okay, DNA has been known for about 2 centuries around, known about. So we need to start moving forward, okay? So that's it. See you later.